Good morning, microbiome nerds. How are you? This is Tracy, and we are starting our session here, and I will be going over just the concept of what the pharmaceutical model is currently in microbiome. So I'm just going to take the first slide here on the presentation. We've got 30 minutes before the advanced microbiome Zoom meetings start. So this is really just kind of a warm-up, get our minds going. And what I wanted to talk about here is when we're looking at what is really known in microbiome science. Really for the last 40 years, what we thought we knew about the microbiology of the gut, we find out now is mostly false. And it was the last 10 years where the Microbiome Project, which is a global endeavor, and the American Gut Project really enlightened us with what is going on in the microbiome. So we have a picture here of the Amazon forest, and what we're trying to depict here is that this is a very complex ecosystem. We probably have just touched the surface of what there is to be known about the microbiome. And currently, when you have, a variety of tests, if we're talking about how the functional medicine world works, we're really looking at, we have a series of companies that will test, and then we have a protocol to follow through. And what's currently happening is that you'll get a laboratory test back, and it will say, okay, we think we've identified a pathogen. It's right here. And then you have any number of nutraceutical companies that will give you a protocol for that pathogen with killing agents, detoxification, and a series of steps. So we go in there to blast out this pathogen, and really this is no different than the BigAg model of taking a bunch of Roundup and throwing it at an environment where you kill everything that it touches, and you may or may not kill that pathogen, but you certainly will develop resistant pathogens or weeds. And what we're finding out now is that the initial identification of that pathogen was really more or less just a guess. As an example, the other laboratories will take you down to the family of what this is, and they may say, okay, this is a Escherichia coli, but there's any number of strains of Escherichia coli. Some of them are commensals. They're perfectly normal in our environment. Others, like hemorrhagic Escherichia coli, those are bad. But they don't tell you which one it is. So that is the problem. So this is what I'm trying to get across, is that what we try to avoid getting into this type of testing, but there's no way to avoid it because the tests out there were technically so bad that Kiran Krishnan really had to search to find what laboratory is being used by the top research centers. And there is really only one that has the accuracy, that has even been awarded certain accuracy awards by the FDA, and that is Cosmos ID. So that in the second hour is what we will talk about, is the difference in that type of testing. But it's really what I'm trying to get across in this very first slide right out of the chute, is that this is a different perspective. We've got to take the 30,000-foot view and say, what do we really know about the microbiome? And the science is very new, and what we have to start treating it like is an organism within itself. It creates its own, and this is what we're going to go into as well, and it's really exciting information as well, is that, remember, we have 10 trillion cells of our body. There's 100 trillion bacterial cells on our body. There are more of them than there are of us. When it comes to the genetics that's required for us to function as a human, if we have 23,000 genes in our genome, but they have 100 times more genes in their genome, and we depend on their genes in order to make our neurotransmitters, make our hormones, metabolize our vitamins, conjugate our amino acids, that we are so intricately involved with these bacteria that we can't live without them, of course, but we have to recognize and appreciate how much of the biochemistry is really up to them. So that's what we're going to go through today, is to show how intricately, woven together our genetics are with the bacterial genetics, how microbiome labs have put together a program to affect this organism of our microbiome and to make changes in it to repair it, and how we can use very specific testing now to look at what the researchers are looking at, the ones that are advancing in this science. And it is advancing very quickly now that we have the type of genetic sequencing that we have. And yeah, that's what I wanted to get across, really just right out of the chute, is that we've got to get away from this pharmaceutical BigAg model and look at it functionally. If you're given a sledgehammer, just because you have it doesn't mean you should go in there and start smashing things with a sledgehammer, when a toothpick is really all you need. And now we're beginning to understand, the intricacies of the microbiome, and we can make big changes with toothpicks, with knowing precisely what you need to do and go after. So that's what I want to open the floor there and kind of get some discussion going so that we can warm up. We'll start seeing a lot of people join us about 8:30 your time, 6:30 Pacific time, 7:30 Mountain time. People will be making sure that they get connected, checking their mics and their audio. So we have from now until, for the next 20 minutes to warm up, kind of get some questions out there. And so I am going to take everybody who's joined us this morning off of mute. Okay. Dawn, I see you there. Laura, I'm not sure where you went, but feel free to check your mic, check your audio. Make sure to remind them to use the high attendance button to fill it out. Yes. So you should have received an email from me that had some links on it. The first link for the first hour is the Total Gut Restoration Lecture. And that's how the company verifies that you attended, so that you can get your discount for the lecture. So make sure you can do that anytime, before, during, after, but make sure you do it before you order. And hopefully these lectures will give you a good idea of, as far as the products that you will get the discount on are going to be the Megaspore, the Mega Prebiotic, and the Mega Mucosa. And we'll go over the protocols for that. And you would be able to calculate, okay, how many people do you want on that protocol, and calculate how many bottles of whichever ones you need. And basically, it's going to take 3 bottles of Megaspore, 2 canisters of the Megapre, and 1 canister of the Mega Mucosa for the program. We have recently launched a 180 capsule Megaspore, and you can certainly substitute that for the 3 bottles. So yeah, that's going to be the protocol there. Do you have any questions about what links to click where or when? I'm going to go back a slide here because there's some links. I also sent out these particular slides. I have 4 presentations. This first one is an overview, and the second one goes deeper into the weeds. And on this first introductory lecture, I have the protocol link, which is right here on the first page. When you click on that, it should take you into a brochure on each of the products so you understand the ingredients, and then it'll go into the titration schedules of each of the products. And here you see, here's the Megaspore ingredients. You see the 5 different species: Bacillus indicus, Subtilis, Coagulus, Licheniformis, and Clausii. Each one of those have a unique and different role in the microbiome. Then you have the Megapre, you've got the ingredients there, the GOS, XOS, and FOS, and what organisms they promote. And then you have the Mega Mucosa, which it's amino acids, immunoglobulins, citrus polyphenols. And then here's where you get into the protocol. And it's, starting with a half cap to 1 full cap every other day. And then the second week, 1 cap every day. The third week, you're up to the recommended dose. And then the second step, what you add to the Megaspore, generally starting at that second month, is a half scoop of the powder. And then the second week, you're up to the full scoop. And then the third month, starting the powder of the Mega Mucosa, half scoop, and then a full scoop. So here you have it showing you the 3 months, and then thereafter, you're really just maintaining with Megaspore 2 to 3 caps a week. Talk about it here. And then here's all the references there. So that is on the first slide. And then also I've got a link here for all of my YouTube videos. I've got, like I said, 45, 46. And you really do need to hear this information multiple times. Okay, so my recording going. You really need to hear it from Kieran. You've got Acacia, you've got myself, and every time you hear it, you hear something different, because at some point you want to be able to present this information as for your community. This is revolutionary. It's unique. It's people are always shocked when they find out that they've been wasting their money on probiotics that die in their stomach, and these products really work. So you want to be able to talk them through, because what you'll find is every single person's microbiome is a different science project, is a different protocol. You start with the foundational protocol, and then you look for how their microbiome organism is going to respond. And then you can do it manually by understanding each of the reactions, side effects, die-off that's unique for that person and know how to respond to that. And now with the testing, you can start off with the test, then put them on the foundational protocol. When you get the test back, then you customize it. Now you're really seeing what is going on in there. And then there's some practitioners who use a combination of both. Some people, they will do manually, and others, they will add the testing and do a combination of both. So I think it's, really, that's how you're honing in your skills when you're becoming a microbiome expert. And I think every practitioner in this space needs to be a microbiome expert. And between the experience of myself and my husband, who is a very gifted practitioner as well, we are here to coach you through the different scenarios until you get really savvy about putting customized protocols together. And so you'll see here, I got 46, some of them very short, talking about multiple different topics, a wide range of different things. But also we have our microbiome nerd meeting every Thursday morning, starting at 8 o'clock Central Standard Time. And, this keeps the discussion going every week, which is important because, it takes time to develop an expertise in this field. And what we've created is a community where you can bring your difficult gut condition cases, talk to a variety of different practitioners. The practitioners that have been coming very loyally to we used to call it the Breakfast Club. We changed the name because people were thinking it was a kind of a social get-together, where it's not. It is a very scientific meeting to strategize protocols. And it's been going on, gosh, I would say about almost a year now. And, we've been through a lot, personally, we had a cancer episode, and we continued our microbiome nerd meeting on trips to, some of the time they didn't realize I was doing hosting a meeting in a different city almost every week, and sometimes we were on the way to a hospital, or we were sitting through a chemo session, or in the real cold days, they didn't realize I was doing some of those still in my bed. But the point is, we're very loyal to keeping the group going, keeping the networking going, and really deepening those relationships so we feel comfortable bringing our cases, our clients, our patients' issues to the floor and resolving them together in some cases. So that, and we've also been able to talk through issues about the products, things we don't like about the products, things we want to see changed about the products, how we want the expansion of the SKUs. So that happens every Thursday morning. But I would hope that, especially after these next 2 lectures, we've got a really good turnout. We've got a lot of people that registered for this. So my encouragement to them will be, take advantage of Ken and Mai's experience. Book a standing meeting, a weekly meeting, in addition to the nerd meeting, so that you're using us as mentors. and work through the different cases and, you know, get to that point, you know, this material front and back. So that's our invitation to you, and we'll extend that to everybody. And I think those individuals that are movers and shakers will take us up on it. So that's what we'll, you know, put out there. Okay. Let's see if anyone else has joined us. I'm surprised that Dr. Kevin, this is the first time he hasn't joined us. So he'll be joining us though later. He's okay. So I think we had a good warm up here. Got to kind of go over the concept and this whole paradigm shift on microbiome testing and protocol strategies. And so I'm going to go ahead and end the recording on this, and then we'll start it up again in about 9 minutes.